Genetic diagrams such as Punnett squares show you the predicted ratios of different phenotypes. They do not tell you the actual offspring because every time gametes meet, they are carrying a unique and random mixture of genes. You can only see the expected ratios of phenotypes if you carry out lots of genetic crosses. That is why plants and animals that breed fast produce lots of offspring while doing so are widely used to study genetics. Direct proportion and sample ratios. Using Punnett squares, you can work out the proportion of an offspring of a genetic cross and expect to have a particular genotype or phenotype. You can also work out the ratios of genotype or phenotype to another. Here we see two Punnett squares. One is of a genetic cross between two heterozygous black mice, which means they have black and brown alleles. The other is a cross that we saw previously. It's the one where a brown parent is crossed with a heterozygous black parent. In the first cross between the two black heterozygotes, we can work out both the genotype and the phenotype of the offspring, and we can work out the proportions and ratios between different genetic combinations. If we cross gamete A of parent 1 with gamete A of parent 2, we have a homozygote black offspring. If we cross gamete A of parent 1 with gamete B of parent 2, we have a heterozygous black offspring. If we cross gamete B of parent 1 with gamete A of parent 2, we have a heterozygote black offspring as well. And if we cross gamete B of parent 1 with gamete B of parent 2, we have a homozygote brown offspring. So the genotypes are, we have 25%, or 1 out of 4 times we will get a homozygous dominant. Half of the time, because we see this happening 2 out of 4 times, so 50% of the time we are getting a heterozygous offspring, and 25% of the time we're getting a homozygote recessive offspring. So when we cross two heterozygotes, these are the proportions or the ratios that we will have. 25% are homozygous dominant, 25% are homozygous recessive, and 50% are heterozygous. This is every time we cross two heterozygotes together. The possible genotypes appear in a ratio of 1 to 2 to 1. 1 being the 1 out of 4, 2 being the 2 out of 4, and 1 being the 1 out of 4. So the ratio of homozygous dominance is 1. The ratio of heterozygotes is 2, and the ratio of homozygotes recessive is 1. As for the phenotypes, we have the heterozygotes as well as the homozygote dominants are all black. So 75% of the offspring are black, and only the homozygote recessives are brown. So 25% of the offspring are brown. These are the phenotypes of when we cross two heterozygotes together. The proportions and ratios of possible offspring will be the same for every heterozygous cross you look at. Every time we cross two heterozygotes, we're going to get these ratios, 1 to 2 to 1, of homozygote dominant, heterozygote, and homozygote recessive. These are the genotype ratios. As for the phenotype ratios, we're going to get 75% of the time the dominant phenotype and 25% of the time the recessive phenotype. So this is a pattern we can memorize to keep in mind instead of having to perform the crosses every single time. If we see that there is a cross between two heterozygotes, we can directly apply these ratios.